I'm Jonathan Munn from the University of Illinois at Urbana-Champaign at EB 2014 ASN poster sessions, here to talk to you a little about my research. The focus of my research is on the effects of hypoglycemia in the ventromedial hypothalamus and how changes in glucose metabolism in the VMH produce changes in counter-regulation, the ability to respond to hypoglycemia. Our treatments were basically a single episode of hypoglycemia, recurrent episodes of hypoglycemia, or saline control in rats. And from these treatments, we took a couple approaches. We used RNA-seq and RT-PCR to look at gene transcription changes, and GC mass spec and LC tandem mass spec to look at changes in metabolites. And the hypothesis that we wanted to test was that glycogen metabolism is changed in response to hypoglycemia. And what we find is that in the VMH, these changes reflect increased turnover. We hypothesize that with recurrent episodes of hypoglycemia, glycogen metabolism may provide protective effects locally in the hypothalamus, but may result in impaired counter-regulation. And so what we see in this figure here is that after single episodes of hypoglycemia, there's an increased synthesis of glycogen, as reflected by increases in glycogen synthase, phosphorylase kinase, and UDP glucose pyrophosphorylase. We also see that there's an increase in glycogen mobilization via glycogen phosphorylase, and even after 24 hours after 3 recurrent episodes of hypoglycemia, there's an increased capacity for glycogen metabolism. And so we tested this hypothesis, but using our RNA-seq, we expected to be able to produce many new hypotheses because of the number of genes that we're identifying. And one of these genes of interest was arachidonate 15 lipoxygenase, which in this figure shows that there's a decrease in lipid metabolism via this gene in the acute treatment, and a big increase after recurrent. Now, arachidonate 15 lipoxygenase is an enzyme that is associated with metabolism of phospholipids, polyunsaturated fatty acids, and other complex substrates. And this change in arachidonate 15 lipoxygenase is reflected well in changes in biochemicals. We identified a number of different lipid metabolites, in particular lysolipids, which are metabolites of phospholipid catabolism. And what we see is the same trend. We see a decrease in the acute treatment and an increase in the recurrent treatment. We see this with changes in cholesterol metabolism as well. And so these findings kind of reflect that there's a change in glycogen metabolism, and that may contribute to impaired counter-regulation, but this change in lipid metabolism, which hasn't been studied previously, may dramatically change the physiology of counter-regulation. And so this change in lipid metabolism may reflect a change in the physiology. Basically, this change in gene expression and change in biochemical concentration may reflect altered physiology, altered metabolism in the hypothalamus that results in impaired counter-regulation.